we confirm those plasmids by restriction digestion with EQR1 XPA1. And how we confirm, once you have, you isolated from these individual colonies, what you did plasmid isolation. The DNA, basically this plasmid you are isolating from the bacteria, and you do restriction digestion of these plasmids with EQR1 and XPA1. You remember why we are doing this? Because we ligated our insulin gene with the vector with EQR1 XPA1. And after digestion, what we do, the final step is we do agarose gel electrophoresis. So these plasmids from these bacteria, they are digested and they are run on the agarose gel electrophoresis, and there you will see, two bands. One will be your vector, the other will be your insert. And if you find on the agarose gel, let's say the size of the one fragment of the insulin gene and then the vector, two bands appearing on the agarose gel, you have successfully cloned insulin in a bacterial plasmid. The next step is you then perform the expression analysis. One thing you should remember, that when we choose these cloning vectors, remember our original question was we wanted to express insulin gene. So our vector, it must have a promoter, a bacterial promoter. Normally in bacteria we use this, inducible promoter, lactose inducible promoter, and there are different strains of bacteria, very efficient strains, which once you confirm your plasmid, then you go to those expression strains. Remember the cloning strains, the E. coli, strains, DH5 alpha or XL blue 1, etc. These are names of the normal E. coli strains which are used for just cloning, not expression. For expression, you have to go to different bacterial strain. Okay, and once you confirm your plasmids, once you have confirmed that your insulin gene is indeed in your bacterial expression plasmid, you then, after confirmation, you go and add inside the bacterial expression strain, and there we have lactose inducible system, and we add lactose in the medium, and insulin will be produced in the bacteria. This is now the production part, the third part. So you remember I told you in genetic engineering you have design part. First we went through whole designing, selected the gene according to our hypothesis, according to our question. We selected vector, we decided host, the whole design of the construct. I just told you, you'll have to have your bacterial expression plasmid, which promoter will be there, which strain you will use. So it involved then when we talk about the construction part, and that's why the whole thing is referred to as genetic engineering, because you have used building blocks, bacterial promoter, human gene, put this chimeric construct or plasmid together in a bacteria, and then bacteria help you to confirm that you are indeed successful in making this chimeric construct. Once you confirm this chimeric construct, you go back to bacteria, different strain of bacteria, and that bacteria is then going to produce this insulin for you. and you will have a mass scale or large scale production of insulin, or let's say any protein. I'm now going away from insulin. Your question could have been production of a different protein which can be used as a diagnostic toolkit in ELISA, or a protein which can be used as vaccine, whatever your questions are. So the whole concept of genetic engineering, and it relies on all the tools and methodologies of recombinant DNA technology. Recombinant because you're combining different molecules of DNA of different origin together. And you have, as I said, you have different strategies to clone this gene. The simplest one in my life, what I experience, is PCR-based cloning, which we will do when we talk about, the next module. So in next module, we will take, let's say, we'll take example of, in the context of present day crisis, like, the coronavirus crisis. In the next module, next part of this module, we'll take, let's say, coronavirus, and we say, okay, we are interested in, generating coronavirus spike protein to generate an ELISA kit. Or let's say another friend of yours is working in a fundamental research lab, in a cancer biology lab. Their lab has done some screen for tumor suppressors genes, and they found some novel genes involved in suppression of cancers, and he's interested in cloning a tumor suppressor gene. But the methodology, tools, everything will remain the same. So that is all about the genetic engineering, and all these tools and methodologies, they will be, we will reinforce them again and again in the next part as well, when we will talk about PCR-based cloning, because this part was just about an intro about genetic engineering. What is genetic engineering? And in the next part, we are now going to have PCR-based cloning, and there we will go through each step in great detail. Okay? So stay tuned and watch the next part.